This is the introductory webinar on the updated MMRRC strain genetic quality control, or GQC, process. The goal of performing GQC is to ensure that tools and resources used in biomedical research are as advertised. This is particularly important for laboratory mice strains that are defined by a specific allele and a known genetic background. The MMRRC's primary mission is to archive and distribute mouse strains carrying an allele of interest. The alleles are the reason researchers use these strains. Therefore, the primary goal and first step of GQC at the MMRRC has always been to ensure that these strains carry the corresponding allele. MMRRC centers always validate the presence of the allele in the strains they distribute. This is the foundation of MMRRC GQC and continues as we enhance the process. A recent advance in the MMRRC strain GQC process, described in our science article, is the validation or the characterization of the genetic background present in a given strain. The MMRRC strain GQC process achieves this goal with remarkable accuracy using state-of-the-art platforms and bioinformatic pipelines. GQC is a necessary step in rigorous and reproducible research. It may also play a role in experimental design, sample size estimation, and establishing baseline expectations for replicability between studies. From a biological point of view, GQC is critical because genetic background may determine the presence and severity of the phenotype associated with a given mutation or allele. Two early examples published in Cell and in Science are shown here. Hundreds of similar examples have since been reported in the literature and form the basis of modifier mapping studies. A great example of the influence of genetic background is provided by a cancer model known as polyomma-T. FVB mice carrying a transgene encoding this protein develop cancer and eventually metastases, but the severity of the phenotype depends on genetic background. This is illustrated in a screen in which females from 27 inbred strains were crossed with males of polyomma-T cancer model to create F1 females. These F1 females carry the identical transgene and paternally inherited FEB genome, and vary only by the maternally inherited genome. As shown in the figure, the average metastatic burden varies widely due to genetic background. This has broad implications for replicability of prior research. Background-driven phenotypic variation is also critical when modeling human disease. This has been the first of 5 webinars explaining the MMRRC strain GQC process. Others include how to read and interpret a strain GQC report, strain classification, genetic replicability, and GQC in the collaborative cross. Additional webinars will be created and posted on the MMRRC website. We would like to thank our funding sources and everyone involved in the MMRRC strain GQC project.